So, good evening, guys, and I am Rishabh. This is a doubt clearing session basically for the course Experimental Biotechnology. So, I would request everyone in the lecture to please ask if there is any doubt in any of the lectures that have been taught till now. So, if you guys have any specific doubt, please specify and then I can explain that. You guys can ask anything you want to discuss from week 1 to 12. Or else we'll start with the question answer session first. So I have made some questions. So further we can in the towards the end of the lecture we can discuss if there are any doubts or not. Okay. So this is the first question. Which of the following techniques can be used to separate proteins from a mixture of proteins based on their molecular weights? So the options are ion exchange chromatography, size exclusion chromatography, SGS page, or sucrose density gradient centrifugation. What can be the correct answer? Okay. Only third. Other options can also be correct. So please give a thorough look. Okay. Any more answers? Okay. Please read the question carefully. So now you guys have studied majorly all the techniques that have been used or are used in the analysis experiments. Okay. So be it cell biology or protein structure, their charged groups, etc. Okay. So we whenever you see the term molecular weight and separation of proteins on the basis of molecular weight is done only in basically two techniques that we have studied right now. First is the electrophoresis, and the second one is the chromatography. So, in case of electrophoresis, to be specific, we have page. So, we have two kinds of page, that is native and the SGS one, SGS page. Okay, so both of them can be used to separate a mixture of proteins based on their molecular weights. SGS page is a denaturing one and native page is a non-denaturing one. Okay, In case of chromatography, only one chromatography helps us to separate the proteins on the basis of their molecular weights, that is size exclusion. Okay, this is basically more of a, sorry, this is basically more of a electricity based technique, and this is more of a gravity based technique. Okay, so both of them can be used to separate the uh, proteins on the basis of their molecular weights. Okay, just a second. Okay, so the correct answer is both 2 and 3. Ion exchange chromatography cannot be the correct answer here, because in that case we use the charge on the protein. Okay, the cumulative charge on the protein as one of the features to identify its properties. Okay, now my question is whether we can use the sucrose density gradient centrifugation for separation. Just give it a thought. I can give you one hint, because we have used cesium chloride centrifugation, density gradient centrifugation, in the separation of the DNA. Okay, do you guys remember where? Have you guys heard of the semi conservative method of DNA replication? So, how it was deduced? Using the heavy and the light nitrogens. N15 and N14 was used in that case. Okay, so the authors Michelson and Stahl, they basically used these isotope of nitrogen, and they separated the heavy and the light band of nitrogen using a cesium chloride cesium chloride density gradient centrifugation. Okay, so that is also a weight based method to separate the proteins on their molecule on the basis of their molecular weights. Okay, now, but the question arises whether we can use this density gradient centrifugation for the analysis of molecular weight. Okay, so the answer is no, basically. Although it can separate the proteins that have different molecular weights, okay, but we'll not be able to identify the the correct way how, or the molecular weight of the protein that we have to analyze. Okay, but in this case, the in ion exclusion, oh sorry, in size exclusion or the SGS page, we can identify the particular protein that we have to analyze, or the specific protein that we have been using. Okay, so that is why density gradient centrifugation cannot be the proper way of separation of proteins on the basis of their molecular weights. Okay, so do you guys have any doubt in this particular question? Okay, so I'm moving forward. Okay, yeah. So, like in case of a sucrose density gradient centrifugation, like in case of a page or a chromatography, we must have a base. Okay, that basically helps us to provide the solution in which the protein samples or the samples can move. So, we are providing a base, that is sucrose. Now, in that case, if we add a mixture of proteins in this sucrose gradient and run a centrifugation at particular rpm or for particular time period, then in that case it will help us to separate the proteins based on their molecular weights. The heavier would reach the bottom and the lighter would reach the top. Likewise, the similar technique have been used by Measles and Install to prove the semi-conservative method of DNA replication. Okay, so based on that, we can separate the proteins, but here what the major disadvantage is, we'll not be able to differentiate which protein is what. Okay, if we know the exact concentration or the exact basically components of the protein mixture that we are adding, then we'll be able to identify, okay, that this band is correspond to this particular protein. Okay, so but in that case we'll not be able to identify the unknown proteins. So that is a major disadvantage for the density gradient centrifugation, although it can separate the proteins on the basis of their molecular weights or the sedimentation coefficient to be specific. So am I clear, or shall I explain more? Okay, so let's move on to the next question. With respect to SGS page, which of the statements is or are true? So what can be the correct answer? There can be multiple correct answer also. So please read the question carefully. Okay. Any more answers? You said 2 and 4. Okay. Any more answers? Okay. Okay, let's start. So, w- okay. So, in case of SGS page, so firstly, SGS page is only done for proteins, not for DNA or RNA. It is a denaturing system in which we add SGS that imparts the negative charge, and we add beta mercaptoethanol that basically is a reducing agent. So it breaks the disulfide bond. Okay. Now up we add simultaneously bromophenol blue, that is a tracking dye. Okay, for visualization, we generally add Kumasi brilliant blue or we do the silver staining. Fine. Now we'll see the options. So ETBR is used to track the progress of electrophoretic mobility. No, this is a wrong answer. First of all, ETBR is generally used for visualization of the DNA or RNA. So the track is always measured by bromophenol blue. So this is the technically wrong answer. Now moving on to the second one, beta mercaptoethanol is used to reduce the disulfide bonds. So yes, this is the correct answer. Then the protein migrates towards the anode. Okay, so now what kind of electrode is anode? Is it a positive or a negative charge? So anode is basically a positive charge electrode and cathode is the negative charge electrode. Okay. Why this? To remember this, you remember anion or cation. Okay, anion is negatively charged. It will migrate towards anode. So A. Then cation is positively charged. It will migrate towards the negative charge. That is, cation will always progress towards the cathode. Okay, so to remember this is easier to remember. Okay, so now the protein, because it's negatively charged because of the SGS, it will move towards the anode. So this option is also correct. Now, the fourth option, the lower molecular weight protein migrates slower than the larger molecular weight protein. Okay, so now whenever we add a sample to a gel, be it a agarose or a page gel, what will happen? The mixture of proteins that we see, in that case, the smallest protein will move the farthest. Okay, so it will not move slower, and the larger protein will remain nearer to the well where it has been added, because it has large size, so it will not move much distance. Okay, so the fourth option is also wrong. So the correct answer is option number 2 and 3. Okay, now the fourth option that I can see is correct in case of size exclusion chromatography. Okay, just give it a thought. Whenever we add sample in a size exclusion chromatography, the reverse happens as in case of the electrophoresis. The smallest protein will travel the longest distance in case of the column. So it will move out the last. And the largest protein will travel the smallest distance and will come out the first. Okay. So this is the case with the size exclusion chromatography. So I hope you guys understood the question. Okay, so let's move on to the next question. A globular protein of molecular weight 50 kilodalton exists as a mixture of monomers and dimers in the solution. The most appropriate technique for the separation of these two forms of the protein is, what can be the correct answer? This is a easier one. Okay, wait a minute. So, what can be the correct answer? Okay, please read the question carefully. So here the correct answer is gel filtration chromatography. Okay, because in case of gel filtration chromatography, the proteins are separated on the basis of their molecular weight. So in case of monomer, if it's a 50 kilodalton protein, so it can be a monomer and it can be dimer. So considering 50 kilodalton to be the size of the dimer, okay, so what will be the size of the monomer? It will be 25 kilodalton. Okay, so there is a huge difference between the two. So if we separate the protein on the basis of their gel filtration chromatography, and if the threshold is around 70 kilodalton, for example, the molecular sieve that we are using is 70 kilodalton, then the 50 kilodalton protein would come out first. That would be the dimer, and the second one that to come out will be 25 kilodalton protein, that will be the monomer. Okay, so is the explanation clear? Okay, so let's move on to the next question. So this is a calculation based. Okay. Hmm. So KD is the distribution coefficient, right? So whenever KD value, so KD is inversely proportional to the molecular weight every time. So whenever the KD value is less, that means the molecular weight is more. Okay. So that is the case. So in that case, the KD value for the dimer one would be less, and the KD value for the monomer one would be more. So am I clear? Hmm. Okay. So this is a calculation based question. So you have given a zero point 0.1 molar solution of glucose, that is a stock solution, and you have to prepare zero point 0.5 ml of the 0.005 molar glucose solution from this. So what can be the correct answer? Please calculate. These type of calculations are routine in a lab experiment. So you guys must know how to calculate these things. Just 1 minute. So do you guys need a hint, or shall we discuss? So in these kind of questions, what we do is we have a formula that is C1V1 equal to C2V2. Okay. So this is for the stock that we have, and this is for the working. Okay. So one answer we got is 0.025 ml. Okay. Hmm. So stock means that will have higher molarity or higher concentration. So this is that we prepare some amount, and from that we can prepare smaller amounts of working stocks. Okay, that will be directly used for the experiment. Okay, so now here we have the stock solution of zero point 0.1 molar, and we have to take out some volume from that to make zero point 0.5 ml of 0.005 molar glucose solution. Okay, so now to calculate V1, we have this. Okay, so now after solving this, the volume comes out to be this ml. Okay, so how will we make this solution? So first we'll add this much amount of glucose solution from this particular stock, and then we'll add water, the remaining amount. So zero point 0.5 minus 0.025 ml will be the amount of water that will be added to form this much concentration of glucose and this much ml. Okay, so is the explanation clear? And every time, if this kind of concentration based or the values based question, you must remember the units in which we have to express. Okay, sometimes there are situations when the answers are given in different forms of units, and we have to identify that. Okay, so from next time, whenever you write an answer, you write answer with the correct units. Okay, that will help you to clear yourself as well. Okay, so let's move on to the next question. This is also a calculation based question on the PI, that is isoelectric point of the amino acids. So we have the C terminal carboxy group and the N terminal amino group in amino acids, and that have the dissociation constants of 2.2 and 9.2. So these are the pKa value of the carboxy and the amino terminals. Okay, so the pK of side chain carboxyl group of glutamic acid. So glutamic acid, as you guys know, is an acidic amino acid. So it has an extra COOH group, and the amino acid lysine have a extra amino group because it's a basic amino acid. So the pK value of the side chain carboxyl group of glutamic acid is 4.2, and the amino group of lysine, that is extra, is 10.2. So now you guys have to calculate the isoelectric point, difference in isoelectric points of lysine and glutamic acid. Okay, so please calculate and then let me know the answer. I hope you guys know the formula to calculate the PI, that is the isoelectric point. Shall we discuss or you need more time? Okay, so whenever we are calculating pi, that is isoelectric point, how we calculate, there are nearly 2 to 3 pK values for an amino acid. Okay, so the nearest ones we generally take an average of both. Okay, so for example, if there is an amino acid that is hydrophobic, so it would have two pKs, that is for the amino group and for the carboxyl group. So we take average of both and we'll get the answer. But in case of acidic or basic amino acids, we have pK three pK values. Okay, so in that case, the nearest ones we'll take an average of both. Okay, so in case of glutamic acid, we have three pK values, this and this. So we'll take a average of these two and we'll give the pi of glutamic acid. Okay, so that would be 6.4 divided by 2, that is 3.2. So this would be the pi of glutamic acid. And for the amino group, lysine, we have these two pK values that are nearer. Okay, so this would be 19.4 divided by 2. So that will be 9.7. So this is the pi for lysine. And then we have to take the difference between the two. So this would be 9.7 minus 3.2. So this would come nearer to 6.50. Okay, so the correct answer is 6.50. So is the explanation clear or shall I explain again? Okay, so whenever we calculate the PI formula is fixed. It is the average between the two pKa values. Okay, so if in case of hydrophobic amino acids like glycine, alanine, leucine, isoleucine, in that case we have one carboxyl group, one amino group, and then we have the side chain that is hydrophobic, so that do not have any charge. Okay, so we take the average of the pK values of these two groups. But in case of acidic or basic amino acids, we have three pK values, that is for the COOH group, the amino group, and the side chain that can be COOH or the amino group. Okay, so this would have one pK value and these two pK values. So how will we take the average in that case? So in that case, we'll see which group has the pK value that are nearer. Okay, in case of acidic amino acid, the COH group will have the nearer pK value. So we'll take the average in that sense. So the pi will purely depend upon the carboxyl group. And in case of basic amino acid, we'll take the pK values of the amino groups. That means the pi in basic amino acid will purely depend upon the amino groups. Okay, so here we calculated the glutamic acid pi on the basis of the carboxy terminal group. Here the pK value of the original carboxy terminal was given 2.2 and for the side chain it is given 4.2. So now if we take the average, it is around 3.2. So this is the pi for the glutamic acid. Now similar case with the lysine also. So this is the amino group pK, and this is the pK for the side chain lysine, side chain amino group of the lysine. So when we take this average, this comes around 9.7. Okay, so now the whole basic question is sorted. Now we have to calculate the difference between the isoelectric point of lysine and glutamic acid. So we'll take the difference between this and this. So that comes out to be 6.5. So now the explanation is clear. Okay, so let's move on to the next question. So this is related to the hybridoma technology. So which one of the following properties of the myeloma cells is used in the hybridoma technology to generate the monoclonal antibody? So what can be the correct answer? And for your reference, I have kept one image also. Okay. Any more answers? Okay. Just 1 minute. Okay. So in this case, the correct answer is option number 4. Sorry, this would be 4. Okay, because the myeloma cells that we are using do not have HGPRT. So that blocks their salvage pathway for DNA synthesis. Okay, and we are blocking this thymidine kinase using aminopterin that blocks the de novo pathway. So basically, only the myeloma cells that exist in the medium would be killed by this uh, aminopterin. Okay, so here we are utilizing the property that is lack of hypoxanthine guanine phosphoribosyl transferase. Okay, so this is the correct answer. So let's move on to the next question. Okay, so match the following illusion conditions in group 1 with group 2. So group 1 refers to the illusion conditions and group 2 refers to the different chromatography. So what can be the correct answer? You have to match the following. Okay, let's discuss. So this phenylcephalose is a hydrophobic interaction chromatography. Okay, chromatofocusing is a chromatography modified variation of isoelectric focusing. Okay, DAEAE Cepheryl is an ion exchange chromatography, and NINTA is affinity chromatography. Okay, so in case of increasing concentration of salt, what we do is we generally separate in case of ion exchange chromatography. Okay, so this would be the third option. So this one, increasing concentration of histidine. So as you guys know that the his tag that we have in proteins is used by the NINTA. So here the histidine will be used to purify this particular proteins having the his tag. So the correct answer is fourth one. So Q is labeled with 4. Then decreasing concentration of ammonium sulfate will be linked to hydrophobic interaction chromatography. So in that case, we have salting in and salting out procedure of the protein. So here it will go with R1. And the last one, decreasing concentration of hydrogen ions, that will be linked to the chromatofocusing. So because it uses pi, that is the isoelectric point of the protein. Okay, so S will be second. So the correct answer is option number 1. So is the explanation clear? Okay, so let's move forward. So this question is also analysis based. So we have four proteins PQRS having molecular weight 60 and 45 kilodaltons and different pi values. Okay, so what kind of gel we can use, page gel, to separate all the proteins? Okay, so whenever we came across any such kind of questions where we have to separate the proteins, so we have to look a broader look. Okay, so because here the pi is 4, pi is 8, 6 and 7.5, so it has a broad range. Okay, so it will go around from 3 to 10. That would be the ideal range to separate all the proteins. Okay, then we have the 4 to 7 range. It will not cover all the proteins because here it's 7.5, so it can, like, between 6 and 7.5 there is not much range. Okay, and it will not cover the 8 one. So this range cannot be used for the separation of the proteins. So option 1 and option 4 technically goes out. Okay, so here we'll be using the 3 to 10 wala range for separating the proteins of pH. Now we'll look at the molecular weight. So in this case, because the molecular weight is large, so 60 kilodalton and 45 kilodalton is really large proteins. Okay, so we'll be needing pore size that much so that the proteins can pass through it. Okay, so if we use more concentration of page, that means the pore size would be less. So it's an inverse relation between the page gel or the concentration of the gel and the pore size. So whenever we are using high amount, that means pore size would be less. So here, because this size of the protein need to be separated, so in that case we'll use less amount of page concentration, that would give us the more pore size. So here, out of these two, 12% gel of the protein will be used. So correct answer would be option number 3. Sorry. So, any doubts in this question? Okay. If not, then we'll move on to the next question. So, an enzyme was purified using ion exchange chromatography, and the results are shown in the table below. Which of the following then 10% would be the ideal choice. Okay, because in that case, every kind of protein can be separated. The 12% one is also higher, but out of these two, 20% or 12%, 12% is the ideal choice. But if 10% would be there, the 10% would be the ideal choice, but not less than that, because the pore size also needs to be sufficient amount. Okay, so here you have to check for the uh, fold purification of the enzyme and the yield of the enzyme after the purification. So please calculate, or if you guys don't know, then let me know. I'll directly explain. Okay, for these kind of questions, we generally calculate the specific activity. That is the major distinguishing feature of the enzymes. Okay, so that is calculated by the total activity that we are getting divided by the amount of protein. Okay, so the specific activity will calculate. In the first step, the specific activity will be 2, and in the second step, it will be 100. Okay, so as the purity increases, the specific activity is also increased, because the impure substances or the toxins or the inhibitors are basically diluted out. So when the specific activity increases as the steps of purification increases. Okay, so here, if we have to calculate the fold purification, then we use the then we use the specific activity for that, because that tells us how much purified our protein is. So it can increase the specific, we can increase the specific activity by adding more number of steps and furthermore. Okay, so fold purification is basically the specific activity at the last step or at the step that we are analyzing, divided by the specific activity at the first step, because then we'll know that how much our protein has been purified. Okay, so here it would be 100 divided by 2, so this is 50. So now at this particular step, after the ion exchange chromatography, our protein has been 50 fold purified. Okay, and in case of yield of the enzyme, we use the total activity. Okay, so in that case, what will be the yield here? It will be 200 by 800 into 100. So that will be around 25%. Yeah, that is not a problem, because here in 10 ml of the 10 ml of the mixture, we have more protein molecules. So, for example, you are isolating protein X. So, in that case, if the last step have around 10 to the power 4 molecules of protein X, but in the case of extract, we have multiple proteins that are present in a cell. Okay, so now our protein has been purified to this much amount with this volume. So this is a good concentration. So the volume doesn't matter at all. Okay, what matters is what is the specific activity of that particular enzyme, because we are purifying the enzyme from a cell extract. Okay, here the volume doesn't play any role basically. So here the correct answer is option number 1. Okay, so let's move on to the next question. So aspartate residues, that is the aspartic acid, is found in active sites of many enzymes. The pK of the beta carboxylate of aspartate is 3.86. So it is an acidic amino acid. It also has an extra COH group. At physiological pH, this group can function as, so physiological pH is basically 7. Okay, 7.4 to be specific. So what can be the actual function of the aspartate group? What can be the correct answer? So whenever we talk about these cases, we have to check if the pH is less than pK or pH is more than pK. Okay, so if pH is less than pK, in that case it would be positively charged, and if pH is more than pK, in that case it would be negatively charged. Okay, so this is COH. Oh, sorry. So this is COH. Okay, now what will happen if this has pK of 3.86 and we are talking at a pH of 7.4? So in that case, the pH is higher. So in that means the H plus ions in the outside environment is less as compared to the pK of this particular moiety. Now what will happen? It will try to release this H plus ion. Okay, and it releases and becomes negatively charged. So, if a moiety is negatively charged, that means it has more electrons to share. So, it will be in search of all the ions that have electron deficiency. Okay, so it can act as a nucleophile. So, nucleophile are electron rich species, so that can donate electrons very easily. Okay, so it can act as a nucleophile, and as it can act as a nucleophile, so it can attract many ions. Okay, so all the ions that can be added to this particular group, this means that becomes a base or a conjugate base to be specific. So here the correct answer is option number 3. Okay, is the explanation clear? Okay. So this was the last question for today. So if you guys have any doubt, you can ask. Okay, if no doubts, then we'll close the session for today. And all the best, guys, for your exam.